Hello, everyone, and welcome to today's webinar titled Human Monoclonal Antibodies for Emerging Agents. This webinar is part of a Microbiology Virtual Week virtual event. And I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free education credits. Click on the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Dr. James Crowe, the Director of Vanderbilt Vaccine Center. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Dr. Crowe, welcome. You may now begin your presentation, sir.
Me and glad to join everybody today to talk about human monoclonal antibodies. And I think I'm going to emphasize we're having a microbiology event, so I want to emphasize a subset of microbiologic agents that are emerging agents, emerging infections, emerging viruses in particular. And of course, this is on everyone's mind because we're all locked down in some way because of COVID. So it's a very pertinent area for us. So that's what we want to touch on today. You may be living in an environment where there's a lot of discussion about personalized or precision medicine. And the idea there is if you study lots of features in lots of individuals, maybe patterns will emerge, and we can, through systems of discovery, find patterns or pathways that are interesting and important, and then apply those out to large populations. But what we've been doing in the history of biomedical research for probably over 100 years are what I would call generalized medicine studies, or N=1, where we take an individual, we study them very carefully, and we find patterns that we can generalize out to the population. And so this is a little counter to the current mode or fad of much research around precision medicine, but I call it N=1 studies. And the philosophy is if our genes are mostly shared, then the solutions to many of our problems can be studied in a single patient. And this turns out to be true for monoclonal antibody studies. Now, this is a high technology field. The technologies for making human monoclonal antibodies did not exist when I got into science. Even I was doing a postdoc in the early 1990s, and that's when phage display libraries and other techniques started becoming available. And we saw a future where human monoclonal antibodies could be obtained. Of course, Kohler and Milstein got the Nobel Prize for their discovery in 1978 of monoclonal antibodies by hybridoma fusion. But the full implementation of monoclonal technologies, especially human monoclonal antibodies, has only occurred in the last 15 or 20 years. Now, these are now the most common drugs in the pharmaceutical pipeline, and they're major drug categories, but mostly for cancer, immunotherapy, autoimmune diseases, rheumatoid arthritis, things like that. They've not been used so much in infectious diseases, but I think the tide's going to change. And part of that has to do with the new technologies that are coming along. So we're using 10 or 12 different discovery platforms. One of them is a platform or a pipeline in which we string together multiple techniques that have come out of recent innovative programs. So we can take peripheral blood mononuclear cells from a human being who's experienced some infection anytime in their past. Even 100 years ago, we've done this with 1918 flu survivors. We can enrich those cells with paramagnetic beads or flow cytometry, ultimately going into a single cell device. We've used 10x Genomics, Berkeley Lights beacon system I'm going to show you today, and other single cell techniques, or even just flow cytometric sorting into one cell per well can work as well. We use Pacific Biosciences or PacBio sequencing or Illumina sequencing or even other companies, but typically longer reads if you want full length, we're using PacBio. And if we're using smaller segments, we often use NovaSeq. Then bioinformatics are needed to sort through all of this sequencing information. And now with synthetic biology, we can make cDNAs and then proteins, express those antibody proteins, do binding, neutralization, mouse studies, and non-human primate studies. So that's kind of the pipeline. Here's what it looks like. This is a colleague of mine, Morris Aboji from Lagos, Nigeria, and he's one of the very brave physicians who stared down the Ebola outbreak that occurred in Nigeria during the 2014 West African Ebola outbreak. He's a physician, and a person came in infected to his private hospital in Lagos, and he and other physicians took care of that person, but became infected themselves. Morris ultimately survived with just supportive therapy, and has told his story a number of times. He came to our institution at Vanderbilt in Nashville, Tennessee, to give a symposium about his experience not only treating but also suffering Ebola disease and surviving. So he agreed to give us white blood cells we collected by a process called leukapheresis, where we filter all the white blood cells that are circulating at one time. We can do it safely in a hospital environment. And then we have staff who take those into biosafety level 2 containment. Of course, the survivors we're typically studying are in the convalescent phase, are not actively infected. They've recovered, so there should be no virus in their blood samples. So we separate out by gradient the white blood cells. By leukapheresis in the top left, you get a whole bag that might have billions of cells, but we isolate those and cryopreserve them, and then use those cells in antibody discovery that is as rapid and large scale as we can manage. And what comes out that back end are proteins, which are monoclonal antibodies that can be studied essentially as drugs from either a basic standpoint to understand the correlates or mechanisms of immunity to the diseases. But since we're making them from human B cells and they're naturally occurring, they're really good candidates for use in prophylaxis or therapy. And we've converted this into really kind of an industrial scale process in some ways. These are just photographs from my phone. I just walk around sometimes and take pictures of what's going on. So at any one time, we probably have over 1,000 human monoclonal antibodies being made at scale, which would be, milligram or even up to gram levels when we need them for structural studies or non-human primate studies. But typically, we're working very high throughput because not every antibody that you make is an antibody that would be considered best in class. There's many antibodies that you would make to a particular target. We estimate we have seen tens of thousands of antibodies to single viral proteins during a response. There's probably more than that. We don't have an upper bound of what you do, but we sort through thousands during these antibody discovery campaigns to find the best in class antibodies. Another thing that has helped us a lot is next generation sequencing, DNA sequencing, and also synthetic genomics. So these are fields that have matured. NGS really grew out of the Human Genome Project, and synthetic genomics, you can practically make a mammalian organism just by synthesizing DNA and going on from there. So we've been able to do things like sequence the entire repertoire of antibody genes or B cell receptors or T cell receptors in a blood sample. And what's interesting about that is this particular lineage is an influenza virus antibody lineage. And this little triangle is the single B cell that we recovered, and we know the function of this. It's a universal flu antibody that we discovered. But by sequencing the same blood sample essentially to depth, we see all these somatic variants, and this would be a clonal lineage. And we can identify multiple instances of some of those somatic variants, and also we can infer the germline or unmutated common ancestor of these antibodies. And once we know the sequences, we can synthesize them and make them as proteins, and that's part of our workflow. So there's sort of two ways to think about getting things ready for epidemics or even pandemics. One is to think ahead of time and be ready when things happen, so that there's not a scramble and chaos, just to have prophylaxis or therapy available prior to an outbreak. And then the other philosophy is to develop technology to move really fast. So I'm going to start with the idea of thinking ahead of time. And we've actually rolled out a program called AHEAD 100, or Advanced Human Epidemic Antibody Defenses, for 100 targets. And we've taken everyone's list. So various people have lists, the NIH, CEPI, WHO, various eminent bodies have published lists, and we sort of put them all together and said, okay, we have a meta list, and we've been working on making human monoclonal antibodies that would be protective or therapeutic for everything on the list, basically. And on the left, you see families or groups of viruses for which we already have between 50 and up to probably 5,000 human monoclonal antibodies, and we've down selected to individual antibody clones that we think are sufficient for prophylaxis or therapy based on preclinical studies. And some of these are in development or even have been given in human beings. In the middle, we always have campaigns ongoing. These are some of the things we're doing right now. And on the right are targets for which we're either collecting cells or making the antigens or the assays. We have to do a lot of preparative work before we pull the trigger to the campaign, because you need immune donor cells, you need a lot of reagents, you need assays for binding and for function like neutralization, you need small animal models. So there's a lot of prep work that goes in before launching, but nonetheless, we're doing a lot of these campaigns. I put my email address in there in this slide because in an audience like this, there's a very broad community, and if there's something that's not on this list, and of course we have other targets, but you'd like to see on this list, just ping me and we'll talk to you about it. Particularly if you or someone you know has had the target organism that you're interested in the past, that makes it even easier, and we can talk with you about participating in our research. So another area that's greatly impacted this field, not only our work, but the work of many others, is sort of the revolution in the efficiency of structural biology techniques, both crystallography and single particle EM reconstructions, cryo-EM, the microscopes and software, synchrotron availability. All this stuff is coming together to allow us to rapidly get structures of antibodies in complex with viral antigens or on viral particles. And so these images are antibody antigen complexes of antibodies that we've made in the last 4 or 5 years or so, done with many collaborators or by ourselves. So a lot of the influenza work we did with Ian Wilson's group and Andrew Ward's group using crystallography or EM, and filovirus work we did with Erica Sapphire's group, and a lot of the alpha virus and flavivirus work we've done with the Purdue group, Michael Rossman, who's recently deceased, and Richard Kuhn and others. So, and also David Fremont at WashU. So there's a lot of people who have expertise in structural biology, and by coupling the antibody discovery with the structural biology, we can begin to rapidly understand mechanism of action. So that's very helpful. So I'm going to give you some examples of this kind of AHEAD 100 philosophy and rolling out an antibody discovery campaign. The first I'm going to talk about is Marburg virus. So Marburg is a filovirus, and it's very similar to Ebola, and it's not good for you if you get this. So, and this sort of supports the idea I talked about of N equals 1, so getting a solution from one person. So this story is quite interesting. There was a woman, Astrid Joosten, from the Netherlands, who, with her partner, traveled to Uganda on safari. This is a picture of her while she was on her travel, her sort of vacation travel. And she visited a cave in Uganda where fruit bats congregate, and there's about 40,000 fruit bats in this cave, and so she visited to see the animals. And then left the country, went back to the Netherlands, and died fairly rapidly afterwards. And Marburg virus was isolated from her blood. And it turns out that she was not the first woman to go in the cave and become ill within a short time period. This cave was sort of a known tourist site. These are the bats in the top of the cave. This photo is actually from Joel Sartori, who's a National Geographic photographer. He's a fantastic photographer who's photographing most of the animals on the planet. And he also has been to this cave, Python Cave in Uganda, and photographed the Rosetus aegypticus, or the fruit bats. So this is the actual cave where Astrid went in. And it turns out that investigators from CDC and other places have gone into the cave subsequently and demonstrated that 3 to 5% of these bats have Marburg virus in their body or in their blood at any one time. So when the news of Astrid's death came out in the news and the association with Marburg, another person was quite interested in that news report, and that was Michelle Barnes. Michelle and her partner, husband, also like to travel to places to see animals and unusual things, and she had been to Uganda to the same cave prior to Astrid, it turns out. Michelle had become similarly infected. She had no direct contact with the bat, but she flew home from Uganda, became ill. No one could figure out what she had, but a number of months after she recovered with only supportive care and no diagnosis, she read about Astrid's case and basically self-diagnosed herself as having survived Marburg. And the CDC then subsequently tested her serum, and she was Marburg seropositive, which revealed that was what had caused her infection. And she gave a TEDx talk, which is quite interesting, in which she showed images of antibodies that we'd isolated from her blood. And, you know, I recommend, if you're interested, to look at her talk. So from her peripheral blood, we contacted her. She gave us peripheral blood. We made monoclonal antibodies from her B cells, and we made about 50 originally, and a number of those were neutralizing for Marburg virus with the real virus, which we did in collaboration with Alex Bukreyev at UTMB. And in color, you see FABs in complex with Marburg glycoprotein, and each color is a different monoclonal antibody. And some of them come in on the left at sort of a 45-degree angle onto the Marburg glycoprotein trimer, shown in gray. And on the right, there's other antibodies that come in at a very flat angle. These are single particle EM reconstructions that were done by Daniel Murin and Andrew Ward's group at Scripps. And they revealed that the antibodies basically bind to the receptor binding domain, or the inferred receptor binding domain on the glycoprotein. So this is what I mean by discovering the mechanism of action. Now we know these antibodies block attachment. Erica Sapphire took several of these. This is an image from the Scripps imaging group, and Erica Sapphire's group determined the co-crystal structure of the Marburg glycoprotein trimer, which is in the surface representation in purple colors, in complex with one of the antibodies in yellow. The darker yellow is a heavy chain, the light chain is a lighter yellow, and you can see that there's a cup-shaped pink structure near the center, and the heavy chain CDR3, or the complementary determining region 3, or hypervariable loop 3, reaches down into this cup-shaped structure, which is the receptor binding domain. So this is a very good outcome for the research, because this was the first atomic resolution structure of Marburg glycoprotein, which had been elusive in efforts to obtain that structure just in the apo or uncomplex form. But in complex with the antibody, Erica was able to obtain the structure. So then we knew the structure of the glycoprotein, and therefore the identification of receptor binding domain, and we had antibodies that bound directly to that domain, and that's how they work. They block attachment. And furthermore, it's very interesting if you look at the receptor binding domain at high resolution, and these are 3 structures that George Gao put into a cell paper, and on the right is the structure that we did with Erica of the Marburg antibody in complex with Marburg glycoprotein in orange on the bottom. And what we could see was there were aromatic residues on the antibody that reached down and interacted with aromatic residues on the Marburg glycoprotein. Well, it turns out on the left, it was already known that another family member, Ebola virus, Ebola glycoprotein, shields the receptor binding domain with a structure called the glycan cap, which reaches down and interacts with the receptor binding domain, and it does so with aromatic residues. And then George Gao's group in China, in this paper in Cell, then obtained the co-crystal structure of the NPC1 Niemann-Pick C1 receptor for filoviruses in complex with Ebola GP, and that receptor also interacts with Ebola GP with aromatic. So this was the first case I'm aware of in which a viral protein and a human host protein and an antibody all use similar structural and biochemical motifs to create pi-pi interactions to make the ligand to bind. And it also shows that this antibody mimics the receptor, and that's why it's neutralizing. So that one of these antibodies called MR191 has been licensed to a company, mAbs, that you may know of from ZMAP work. So they were very interested in filovirus work. We have studied this antibody in monkey models, which are invariably fatal. So this is a survival curve, and by 10 days, all untreated animals are dead with this lethal challenge, but antibody-treated animals are all living. So the NIH was excited about this and put out a press release, and furthermore, because of the promise of this antibody, the United States has a particular agency that supports manufacturing called BARDA, and you're aware of BARDA from the news now because they're very involved in supporting COVID manufacturing efforts. So BARDA put a significant amount of money in moving this antibody Marburg 191 into manufacturing, and this essentially will be the drug for Marburg that's available. And in fact, there was an episode in a containment lab where a person was exposed to Marburg and needed to be treated and was treated with this antibody on emergency IND. So it just shows you how one person, Michelle, the study of her repertoire and making monoclonal antibodies can lead to a drug. So Michelle, as far as we know, is the only known Marburg person in the Western Hemisphere. And yeah, we could do all that work with just a blood sample from her. Now, so I mentioned the AHEAD 100 program. We don't only want to do Marburg, we want to do as many major pathogenic viruses as possible. Now, to some extent, we've been sort of reacting to things that have happened. You know, there's been bird flus and Zika and chikungunya and Ebola and, now SARS-CoV-2. So we are doing AHEAD 100, but we typically will pivot to do the microorganism that's in the news and is desperately needed for a public health standpoint. So this is a study that we did, published in 2016, of Ebola immune donor repertoires. And when we did that, this is a Circos plot, we actually were looking not only for Ebola Zaire reactive antibodies, but also broad pan Ebola virus, which would be Sudan, Zaire, and Budabujo, or pan filovirus, which would also include Marburg. And we saw reactivity in 3 species pretty frequently. So this suggested you could get single antibodies that would get all species of Ebola, and maybe that you could make a vaccine for all 3. Now, most of the work in Ebola has been focused on Zaire, but we made antibodies that were quite broad. Again, this is single particle EM reconstructions from Andrew Ward's lab on the right, showing that many of these antibodies bind receptor binding domain at the top, but also we got stem or membrane proximal external region antibodies on the bottom. And a lot of these antibodies are very potent. This is a single monoclonal monotherapy using an antibody we made called EBOV 520. And if we give 2 doses in a therapeutic model on day 3 and 6, we get complete survival even using monotherapy, which is a good model of what happens in humans. So we have there are now licensed antibodies for Ebola from at least for use in affected areas, and certainly antibodies that have been proven to be effective from Regeneron, and you may know about the mAb 114 antibody that was developed by Ridgeback and NIH and others. So the principle of using monoclonals is already out there in humans, and in our case, we're now trying to develop next-gen or second-generation Ebola antibodies that are broader, that get not only Zaire, but also Bundibugyo, Sudan, and are more potent and maybe could be given later in lower doses. We also worked on Zika when that happened, so we were already working on most of the flaviviruses, so dengue 1, 2, 3, 4, and West Nile virus and Japanese encephalitis virus, and other flaviviruses. And Zika was on that list, even though it only had been in Africa historically and in the Pacific more recently, but then a worldwide outbreak occurred. So we made potent neutralizing human antibodies for Zika. The neutralization curves are on the top left, and we got antibodies. The pipeline was starting to mature, our experience with it. So we started getting antibodies with single-digit 5 nanograms per ml of half maximal inhibitory concentrations, or IC50s. So it does not take much antibody to inhibit the virus, and that's what you want, a very, very potent neutralizing antibody. When we did structural studies with Richard Kuhn's group, we found that these antibodies are the most potent ones are very unusual. For instance, some of them bridge 2 dimers in the flavivirus, so they basically staple the virus together so it cannot uncoat. We did studies in animal models, including pregnant models with Michael Diamond at WashU, and show that passively administered antibodies to mothers even get into the head of the fetus and protect the fetus brain against virus. And this particular antibody, Zika 117, is now in clinical development with support from various U.S. government agencies and a company called ID Biologics. This is another example. There's a virus called Hendra virus. So there's a whole group of viruses, Nipah viruses. Hendra and Nipah are the 2 that you've mostly heard about. So Nipah has been in Malaysia and more recently in Bangladesh and even Kerala area of India causing outbreaks. So this goes from bats to people, or bats to pigs to people in Malaysia. Hendra is very similar, goes from bats to horses to humans, often even to veterinarians who are caring for the horses, and happens mostly in Australia. So henipaviruses are an important potential cause of epidemics, and they're very lethal. So we worked with an individual who had been inadvertently exposed to a horse vaccine for Hendra. So this was not a human vaccine, but the person inadvertently was exposed to the vaccine and developed immunity to Hendra and Nipah. So we identified a lot of antibodies. The first set was about 42 antibodies, I think. And we've already shown, this is unpublished work, that we get very potent antibodies from this individual that neutralize Hendra virus. Even this is in microgram per ml, less than 1 microgram per ml, we still get nearly 100% neutralization of Hendra. Interestingly, they also, even though they were induced by Hendra, they're very broad in the sense that some of these also neutralize Nipah virus, Bangladesh, and Nipah, Malaysia. We've done co-crystal structures in our lab. Chenwei Dong and our group has gotten co-crystal structures of a number of these antibodies. So in yellow and pink, you can see the FAB, and in green is the Hendra G protein, or the head domain of the G protein. And so this antibody binds straight on to the Hendra G protein, which is now also termed the receptor binding protein. But in gray right here is the actual receptor. So ephrin B2 and ephrin B3 are the receptors. So that co-crystal structure is known. So it turns out, like I showed you before, this is another instance in which The FAB is binding identically on the same footprint as the receptor, and therefore that's how this antibody works. It blocks receptor binding or blocks attachment, and that is a very potent method for neutralization. Now, not all antibodies work by neutralization, neutralization is usually done by mixing a virus with an antibody for something like an hour in vitro and then putting on cells to see if there's any infectious virus left. Some antibodies work by other mechanisms, and by working at scale, sometimes we stumble into these. This is an example of an influenza antibody that was quite broad. It recognizes virtually every influenza A virus hemagglutinin protein that we've looked at. And if you look at the top view of this co-crystal structure that was done in Ian Wilson's lab, you can see that the FAB, so a single FAB was crystallized in complex with what is shown in the ribbon diagram, an HA head domain. But if you represent that complex in the context of the trimer, so HA is a trimer, there's no way that that antibody could bind when the HA is in a closed trimer, because the epitope for binding on the HA head domain is on the inside of the trimer, is in the trimer interface, or the interface between two HA heads at least. And so this is very important because it reveals that at some point during the virus life cycle, the HA head domains must open up. And it turns out when we put this antibody into neutralization assays, it does not neutralize. We do not see any neutralizing activity for the antibody, which might be discouraging, but we put it into animals. We put it in animals, it protected against viral replication, weight loss, and other disease markers. So, and it did that for H1, H3, H5, H7, various subtypes of flu A. So this is an example of a non-neutralizing antibody that works by a non-canonical method. And I think you're going to see a lot more of this. We've looked at this in other virus families, and you're going to see a lot more antibodies to hidden occult epitopes, or what appear to be occult epitopes in the interfaces between protomers that comprise an oligomeric construct. So that's just kind of the snapshots of what we're doing by working our Ahead 100 list. Can we get important antibodies for as many targets as possible, and to do that methodically and make thousands of antibodies and whittle down to ones that we think could actually be prophylactic or therapeutic. But I raised at the beginning, do you want to plan ahead, or what happens if what happens is not already available, is not even on your list? People have sometimes called this Agent X. So we need to be able to prepare for things we know about, but also need to be able to respond to things that we didn't know about or we weren't prepared for. In that case, I think people want to move fast. And there's been remarkable advances in the speed of monoclonal antibody discovery over the last 2 years, I would say. So we were challenged about 3 or so years ago to develop much speedier platforms by our participation in what is called the Pandemic Prevention Platform, or P3 program, which was really a grant call that was subsequently funded by DARPA, which is the Defense Advanced Research Projects Agency. And this agency of the U.S. government does not fund things that can be done. If you can do it, they don't fund it. So they would say, well, the internet came out of DARPA funded research. It was originally called DARPANet, actually, if you remember. And GPS technology came out of DARPA funded work, and stealth materials, and so many advanced technologies came out of DARPA. So they put out a call and said, hey, we want capability to make a monoclonal antibody and put it into human being 60 days after the first case occurs. That was sort of the model. And of course, that was ridiculous at the time. Most vaccine projects historically have taken 20 years or more. Most antibody projects take 5 to 10 years. And even making an expression line, a CHO cell line, typically takes 18 months in the industry. So to say that you would go from an outbreak or a blood sample to injecting someone with GMP material in 60 days was ridiculous, but that's the kind of thing that they do. They put out aspirational goals. So we were selected as one of the 4 sites. The other 3 were Abcelera, Duke University, and AstraZeneca, and all those sites have been participating in this program, including the current COVID effort. And so what we did to learn how to do speed, we were supposed to be practicing. We're supposed to practice 4 times, 4 years in a row, do an all-out effort, which we've called sprints, where you go from start the clock, day 0, time 0, a stopwatch, and then hit the stopwatch when we're done. So our first big practice run was in 2019. So we started in January, and we went from blood sample to finish full protection of primates with an RNA that could be used for clinical trials essentially in 78 days. And so I can just sort of talk you through what that looks like. Our team, I did not agree with this. Our team was excited and fairly confident that they could do this first sprint. The first target was 90 days actually. And they thought, well, we can do this in 90 days. And I was skeptical, but to hold ourselves accountable, we were tweeting this out live. So January 14th, 2019, this is Pavlo Gilchuk on day 0 or day 1, however you want to count it, started sorting in containment blood samples from a Zika immune individual. We use Zika as the model organism for to do this particular sprint. We ended up on the same day in a 10x genomics platform. Then we're using liquid handlers to make libraries from single cells. They went to our core labs. We did sequencing. Our bioinformatics team processed those immediately. We sent those sequences to a commercial partner, Twist Bioscience, been a great partner with us, who made hundreds of antibody gene cDNAs and put them into vectors by Gibson assembly and shipped them back to us in essentially, a week to 10 days. We opened up the FedEx box, and in 96-well format using liquid handlers, we transfected cells within 2 hours of receiving the synthetic genes. And 2 days later, we had recombinant antibodies that we tested for neutralization. Now, Pavlo Gilchuk and our group worked out a new method for testing neutralization. So most neutralization tests take about 5 days, and we couldn't wait that long. In a 60 to 90 day sprint, you can't spend 5 days just doing your primary screen. So he and others in our lab developed a cytopathic effect kinetic reading using a device called Excelligence, which is sold by ASEA. And generally we put cells in and there's electrical sensing of the cell impedance, which measures the intact monolayer of cells. And when the cells fall apart, the impedance goes down. So you can see CPE, the cells are flat, 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 and then they crash because we've added virus. But in this well, flat, 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 the antibody maintained the integrity of the cells. So this was a way in which in approximately 18 to 24 hours, we can see the neutralizing effect in a very high throughput manner. So this is a 96-well plate. There's 384-well versions we're using now. And in doing that, on the fly, we identified lots of new neutralizing antibodies for Zika. We did competition binding on antigen so we could identify groups that competed for each other, which means they're binding to essentially the same antigenic site. So we could sort of bend them into groups, which ones are binding the fusion loop or the domain 3 or the domain 1-2 hinge or quaternary sites. So we knew the groups. We worked with a group, Integral Molecular, has been a good partner with us. They made alanine mutations at every position in the Zika envelope protein, and by looking at lack of binding to individual members of that library, we were able to do alanine scanning during the sprint, so we knew exactly the residues that were important for escape. We did a series of mouse studies with human monoclonal antibodies. Mike Diamond conducted these. I think over a 10 to 14 day period, they studied 500 or more mice. We delivered these antibodies. We made them as IgG protein or as messenger RNA. So the genes encoding the antibody, we did it both ways. The antibodies that we got were so good, we actually had to repeat some of these mouse studies on the fly. This particular study is very stringent. We're only using 0.25 milligrams per kilogram. And you know that in typical mouse studies, you would be using 10 milligrams per kilogram. But all of our antibodies worked at that dose, so we had to keep dosing down until we could discriminate. And even at a very small dose, we saw complete protection. And then Dan Baruch's group, again, during the 78-day period, did a subcutaneous antibody administration the day before challenge, and then challenge with Zika virus. And we looked at 3 different doses of the lead antibody, 2 mg per kg, 0.4 mg per kg, or 0.08 mg per kg, which is 80 micrograms per kilogram, which is extraordinary low dose. And you don't see any Zika RNA in those animals, whereas the sham treated animals had a nice replication curve for a virus. So 100% protection of primates in 78 days. And this work was just published this month in Nature Biomedical Engineering. If you want to see how all that's done, if you want to try it yourself, we tell you exactly how to do this work at scale. And I mean, you might say, well, RNA, what's with that? You know, I've heard about RNA biology, but that sounds a little pie in the sky. Well, of course, Moderna was actually the first COVID vaccine out of the gate using RNA technology in the COVID sprint, and they've done that based on previous studies. But we actually work with them within the context of the DARPA program, a previous DARPA program called Adept Protect, and we made chikungunya virus neutralizing antibodies as part of that effort, and gave them the genes to Moderna. Moderna made them as mRNA, has actually completed a clinical trial that started in February 19 and has gone very well. So this is the first instance of a soluble protein of any kind, but definitely the first instance of an antibody being delivered as an mRNA, and the antibody expressed well. And so this is a promising new delivery technology that allows extreme speed. So I think you're not seeing a lot of mRNA antibodies right now, but you're going to see this in the future, and it's already been in man. So then to transition, we did sort of the fake sprint with Zika, and I kind of recovered, and we were getting all geared up in January 2020 to do another sprint. We were actually going to simulate an avian influenza entering the United States from somewhere in Asia. That was our simulation, and we were just about ready to go to spend a couple months doing that full out. And essentially, right in the middle of our preparations, SARS-CoV-2 happened. And you remember, this is the New England Journal report, which states on January 19th of this year, the first case that was known at the time, the first identified case, was a man who walked into a clinic near Seattle. We decided to pivot and work on SARS-CoV-2 immediately in consultation with our DARPA sponsors. And so by 7 days later, on January 26th, from the time the person walked in, FedEx staff member delivered to my home on Sunday morning in Nashville, Tennessee, a blood sample from the first case. So we immediately launched switching on the fly. And the problem was we didn't have any reagents. We had some blood and no reagents and no assays, and we didn't have any time to prepare. Fortunately, during this epidemic and pandemic, many investigators have released information very rapidly. So the sequences came out of the spike protein, and then many in the field made structures. So Jason McClellan made structures very early on and released them, which revealed the receptor binding domain, and even the receptor binding motif, the N-terminal domain, and the open and closed forms. And it was determined very rapidly. ACE2 was the receptor, and the complex of that was understood. So that gave us a blueprint for what to do. So that was very helpful. So we built assays while we were doing the work on the fly to use that information, for instance, using streptavidin beads, making either the receptor binding domain as a reagent, or the full-length spike protein, or the active domain of the spike protein, and recombinant ACE2 with a tag. And we could build assays by labeling antigen and identifying antigen-specific B cells by flow cytometric sorting. So we sorted antigen-specific cells, spike or RBD cells, activated them in vitro, and then we worked with Berkeley Lights in this case on their Beacon instrument, which was a fantastic collaboration. So this is a high-throughput single-cell device, and the way this works is if we have a bead and a single cell secretes an antibody, and we can detect that antibody captured on the bead, we can see these in single cell cultures essentially. So single cells are deposited and the antibody spills out into a main chamber, almost like a volcano. And so you can sit and watch the live cells making antibodies. Fantastic. And we even developed a neutralization assay surrogate using single cells, which is a blocking of the interaction of the receptor binding domain with ACE2, which simulates the virus binding to the receptor. And so we could demonstrate, and you can see here the little pens with a single cell at the bottom. We could demonstrate that some of the antibodies bound the receptor binding domain but did not block binding, but some of the cells were making antibody that did bind to the receptor binding domain, but the ACE2 did not bind in the presence of antibody. So essentially we did a single cell neutralization for the first time that I'm aware of. Now, I already mentioned that in our workflow you can take single cells by Berkeley Lights. We also did parallel workflow, as I've shown you before, with 10x Genomics, and then again, PacBio or Illumina sequencing. We sent those sequences to Twist Bio, who sent us back again, just as I described. And this is now a 384-well real-time neutralization assay. And again, Pavel Gilchuk worked this out with Seth Zost in our group and was able to do dilutions of antibodies during screening, 1 to 20, 1 to 100, 1 to 50, and 1 to 2,500. And here you can see impedance goes up, and then add virus, and then impedance goes down. And so in this case, these 4 quadrants, these cells were infected. But in this green case, impedance continues to go up, even in the presence of virus, out to 1 to 100, but fails at 1 to 500. So we were able to determine potency during screening at high throughput right off the first expression, which was really fantastic. Cut a lot of time out. So then, because these are recombinant, we can put them into what is ultimately a manufacturing cell line, a CHO cell line, and we screen them not only in the ways that I showed you, but also in ELISA against full-length S protein, against the receptor binding, so a subset bind to RBD, some bind the N-terminal domain, and we also looked at SARS-1 because we were interested in cross-reactive antibodies, so SARS and MERS, and then neutralization assays were done with real virus in biosafety level 3 with Michael Diamond's lab, also with Ralph Barrick's lab at UNC for confirmation. And we see with wild-type virus, we have antibodies that are down into the nearly 10 nanogram per ml IC50 range, so against wild-type virus. And just a caveat that if you use pseudoviruses where you take VSV and you put SARS-CoV-2 on a pseudovirus, the pseudoviruses overestimate the potency. So the same antibodies that are 10 or 100 are less than 1 in a pseudovirus. So I think you need to be careful when you're looking at neutralization assays. Very grateful to the WashU team. This is pictures of them. Brett and Swathy are some of the team members there who worked in the BSL-3 and animal BSL-3 facilities even during the whole lockdown. And we were able to rank antibodies in terms of neutralization, ACE2 binding or blocking, and then binding to RBD or the ectodomain. And by doing all of these assays very rapidly, we could identify potency. We again did competition binding and found various groups that competed, and we could interpret those using alanine scanning again as in terms of where these things were binding on the spike protein. And we even went one step further during this particular sprint, and we took 2 antibody combinations that each antibody recognized the RBD but were not competing for binding. And this is actually a single particle EM reconstruction we did in the lab. Elad Benstein in our lab did this on trimer, and you can see these 2 antibodies can simultaneously bind, and we looked at neutralization as you do dilutions of one antibody and the other together in combinations, we can detect synergy occurring with the two. So this is a really beneficial effect of a combination. They're not only additive and resist escape, but we actually get synergy, which is a desirable property. Mike Diamond and Ralph Berrick's lab have put these antibodies into mouse models, either through adenovirus transduction or transgenics or other methods. And these antibodies work in vivo in mouse models and even against virus or histopathology. And then again, Dan Baruch's lab worked with us on non-human primate studies in which either one of these two antibodies that came out of the sprint were used as monotherapy. And isotype control, you see lots of virus shedding. We don't see a single molecule of RNA in the nasal swab or the bronchoalveolar lavage in either of these monotherapy situations. So these were nominated as frontline drug candidates. And just to kind of think through what I just showed you happen, we got a sample ultimately that gave us the lead candidates. We received that sample in Nashville on March 14th. Ultimately, we did a sprint over a 25-day period and then transferred the sequences to a pharma partner, AstraZeneca, and August 25th, they started a phase 1 trial. We've already published most of the stuff I showed you during that sprint in Nature and Nature Med, if you want to look at that. But a phase 1 trial in August 25th from efforts where we got the sample on March 14th was really pretty rapid. So we're doing more animal studies, accumulating structures. We're working not only with AstraZeneca, but other partners to further some of these. They're being developed in CHO cells, in plant expression, in tobacco, and also as mRNA. These antibodies have been incorporated in diagnostic kits, and vaccine companies are using these antibodies to characterize various lots of their vaccines to show that they contain conformational epitopes. So in summary, what I showed you today is the first half. We can prepare ahead of time by making lists of things that are likely to happen. But in the back half of the talk, I said, well, we can discover antibodies really fast, and these approaches enable us to get into clinic rapidly. So antibodies off of these platforms have already been into people for chikungunya, Marburg, SARS-CoV-2, and others are bumping along in the pipeline. I do think in the future you're going to see mRNA as a delivery mechanism, certainly going to be studied and has promised to work as a fast delivery system. And also you've heard lots about vaccines and antivirals, and I think increasingly what you're going to find is antibodies are going to become the future of preventing and treating infectious diseases. One thing I didn't mention is most of the trials that are being done, certainly with antibodies that we've developed with various partners, have long half-life mutations, and so some of the actual molecules are predicted to have a half-life of 60 to 90 days, but the protective effect could be projected as long as a year or maybe even 2. We'll have to see from the trials. But when you start getting into potential protection for a year, this is going to be competitive with the type of protection you see from vaccines. So increasingly, antibodies are going to be important in this space. And I've tried to mention collaborators we worked with along the way, Diamond, Berrick, Baruch, Dr. Lee's lab at Emory, and Helen Chu at Seattle have worked with us on samples and assays, as Mario Ostrowski, who provided the samples that led to the lead drug candidates. We've worked with some great companies, Berkeley Lights, the beacon I showed you, Twist in the synthesis, 10x Genomics work with us, and these two companies, ID Biologics and AstraZeneca, are developing clinical SARS-CoV-2. We had support from Merck in Europe, Merck KGaA in Darmstadt, Germany, with the Future Insight Prize. Erica Sapphire, Ian Wilson, Andrew Ward, Alex McCraeff, the folks at Integral. The mRNA work we did for Zika was done with IDRI in Seattle and folks at Moderna. And we've gotten funding from DARPA, but also the NIH. And being in Nashville, it's a music capital. Dolly Parton lives here. And Dolly Parton made a lead gift to Vanderbilt to support research early on the outbreak, to make a point of hope, and we benefited some from that. So thank you very much. I'm happy to answer questions.
Thank you, Dr. Crowe, for that informative presentation. And we will now start the Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. just click on that Ask a Question box located on the far left of your screen. Dr. Crowe, we have quite a lot of questions coming in, and we'll try to get as many as we can, and any questions not answered will be answered via email. First question, what about antibody from suspect COVID-19? Tell us a little bit.
Antibody from suspect? I don't fully follow the question. Say it again.
Yeah. What about, the audience member says, what about the antibody from suspect COVID-19? Or perhaps maybe they're saying, do you suspect a certain antibody from COVID-19? Or I'm not exactly sure how the question. Would you like to answer that one via email?
Sure.
Okay.
I don't fully follow that question.
All right. Perfect. Our next question is, what steps do you and your lab group take to assure the safe antibody treatment for pathogens and viruses?
So safety is paramount. We are basically a discovery unit. We do not do manufacturing and we don't do trials. So we license our antibodies out or provide them to various parties, and they're mostly focused on safety. But things that are done, toxicity testing is done in good laboratory environments. It's very expensive. It's typically done in rabbits and primates. Tissue cross-reactivity is examined, so antibodies are put on all sorts of human tissues to make sure that none of the antibodies are autoreactive or bind any tissues. We also try to look at the predicted half-life in especially non-human primates, because you can get anti-drug antibodies, which would make antigen-antibody complexes, antibody-antibody complexes, which is not good for you, and also reduce the half-life. So there are a lot of things that go on during the early development stage after discovery for safety purposes.
Thank you for that. And we're unfortunately getting closer to the end of the time. I just want to ask one more question before we go and give you time to have some closing remarks. What is the difference between a wild type virus and a pseudovirus?
That's a good question. So the viruses that are circulating in people would be field strains. Once they're into a lab and you've cultured them in cells, people call those isolates, and they may change in culture, but typically people also often will call those wild-type viruses because they don't change very much in culture. So that's a virus that you would use in the biosafety level 3. If you take another virus that's not pathogenic for humans or less pathogenic, such as vesicular stomatitis virus, or you could use debilitated HIV, and you could take out the naturally occurring surface protein of the virus and swap in SARS-CoV-2 or some other virus, that becomes a pseudovirus. But usually they're debilitated by deletion of an internal gene, so they can't make more virus. They work as particles. So they can go in a cell and make a signal, but no virus comes out. They're much easier, safer to work with, and we work in a biosafety level 2 lab. We don't work in biosafety level 3 or 4. We do that with partners. So we oftentimes will use these pseudoviruses that mimic the virus, but can be used at biosafety level 2, and that's been very helpful to the field.
Wow, this is incredible stuff, and I just want to thank you again for your presentation. Would you like to provide our audience members with any closing remarks before we go?
I just like to say it's been an amazing experience to work on antibodies. I've been in this field for 25 years, and to see the acceleration. What I showed you today is our work in our lab with many collaborators and partners. But if you look at what's happened with SARS-CoV-2, there's already antibody programs. I mentioned ours with AstraZeneca's in the clinic, but Regeneron, Lilly, Vir, various corporate partners have manufactured antibodies and already gone into trials coming from a variety of sources, and there are efforts in China and in Europe that are similar. So just really, really exciting to see the impact of antibody technologies during this pandemic, and I just can only believe this is going to go faster, bigger, easier, and it's just really encouraging to me that antibodies may be a major part of the solution for epidemics and pandemics in the future. So I hope you enjoy it. I'd love to see more people get into this field. It's a really exciting field.
Thank you so much. And thank you again for your time today and for your important research, fascinating research, really. Before we go, I also want to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via your contact information you provided at the time of registration. And this webcast can be viewed on demand for 6 months, so up to March of 2021. Labroots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, be safe, stay healthy. Bye-bye, everyone.